Hello, I'm Sonja Isken. I'm the Program Director of the MSc Biotechnology. And in this video, I will give the definitions that we use at Wageningen University for Biotechnology and 2 examples to make clear what biotech students do once they study here in Wageningen. Our definition for biotechnology is that biotechnology is the use of organisms or parts of organisms for industrial, technological processes. A typical biotech process starts with a production cell that is optimized via DNA modification after studying proteins and DNA, and then cultivates those cells in large bioreactors, scale it up, make a downstream process where you recycle as much as possible byproducts while you control and analyze all over the way. With this technology, we make different applications possible, spreading from medical and health issues over food towards biomaterials or bioenergy. And the first example that I would like to give is from the medical field. It's a production of a vaccine. And that's done with a baculovirus. The whole story starts only decades ago when farmers had problems with caterpillars who destroyed their plants. And they were looking for an alternative for insecticides that were not in a chemical base and more environmental friendly than the ones used in the past. And this environmental friendly alternative was found in a baculovirus, a very specific virus only killing certain insects and not the insects that you do not want to harm. Of course, you could now cultivate the viruses in caterpillars, but that's not what biotechs do. Biotechs normally use bioreactors and use only the cells from the insects and produce the viruses in insect cell cultures. We scale it up with process engineering techniques to get enough cells and then also enough viruses. That's the way to get a nice biological treatment for the plants. But that's only the start of a nice story. Because the story here now starts because we can modify the DNA of the virus. We can take out a part of the DNA which is in the virus and replace it by DNA, coding for proteins we are really interested in. Then we get a recombinant virus. If we, for instance, add DNA for proteins from a dangerous virus, we get an undangerous insect virus that on the outside has proteins from a dangerous virus. In this way, you can produce vaccines in a very safe way. And this was done at first for avian flu, and then later on also for some cancer vaccinations that are used in the Netherlands. We were busy developing a vaccine against Zika disease when the pandemic for corona started. And then we switched over for the development for a novel corona vaccine. I use this example to show you that you use biological disciplines and engineering together to come to nice applications and nice solutions. You need knowledge from biology, from cell biology, from molecular biology, but also from process engineering. My other example shows that you also can do this for a total different application, and that's the production of bioplastics. Wageningen University has a very long tradition in producing of food, but often we only used parts of a plant for the food production. Think about the grain and the cereals. You leave the straw behind, but in the straw there are very interesting chemical compounds, and this can be used to produce something like bioplastics, and that has been done here in Wageningen. In the straw, we have change of sugars, polymers, and they can be splitted by enzymes. Those enzymes are produced in a biotech way from fungi in large-scale cultivations. And then you can use those enzymes to split the polymers from the straw and get sugars. And to be very honest, not as nice as on this picture. Unfortunately, it's a little bit blobby what we get with different type of sugars. But anyway, there are some microorganisms that can use the sugars to make lactic acid. You know this from dairy industry, where you get the acid taste from the lactic acid bacteria. But unfortunately, lactic acid bacteria are very kind of having certain type of sugars, while we get from the straw really a mixture of a lot of sugars. But other microorganisms, other bacilli, can use all these different type of sugars. So what biotechs do, if they have 2 organisms that can do things that you would like to have in one, you transfer the DNA from the one to the other, you get a modified strain and use that strain. And in that way, we could make a strain that can transfer the different sugars that we get from the straw towards lactic acid. Then it's quite easy to polymerize lactic acid to get polylactic acid, which is bioplastic, which can be degraded very easily in nature. So also here you see an example that uses biological disciplines and for large scale also engineering to come to a solution using biotechnology. If you go and study the Master Biotech, we want you to become an expert in one of the disciplines that we have, either the biological direction, an expert in DNA modification in the cells and the proteins, or an expert in the engineering direction for bioreactors, for downstream processing. You can also focus on certain applications that you are most interested in, only food, only drugs, only bioenergy, biomaterials. But in the Master, you get an expert of one of the aspects, but you learn to use your expertise in a complex problem with experts from the other disciplines. So I hope that this helped you a bit to make a choice for your master program. And if you have more questions, you can send an email to us to mbt.msc@wurt.nl.